So, it's now my immense pleasure to introduce and welcome Professor Clare Mills from the Manchester Institute of Biotechnology from the University of Manchester. And Clare's presentation is Beyond the IFAAM study, What Next for Preventing and Treating Food Allergic Reactions. Thank you very much.
Thank you very much, Tony, for the introduction, and thank you also for inviting me here today to talk about one of my favourite subjects, and actually the Anaphylaxis Campaign. I'm sorry David isn't here. He represented the patients in the first ever project that I led on food allergy. So patients have been at the heart of what we do. So just to declare my interests and the work that I do, and most of what I'm talking about today is actually funded through the European Union. So IFAAM was a project that was born out of the Europreval project. I think we finished Europreval, felt we collected a lot of data but hadn't finished the job. We were very fortunate that there was a call which we applied for and were successful in getting IFAAM, Integrated Approaches to Food, Allergen and Allergy Management, really taking a lot of the data from Europreval but other work forward to develop tools to help the food industry manage allergens in food, but also to address the other part, which is about taking the information and helping allergy clinicians, healthcare professionals, help people with food allergies to protect them from accidental reactions. And how we did that very briefly was we had a number of modules going all the way from nutrition through severity to risk models for the food industry and tools for allergen analysis, and something that was innovative as the Allergy Lab health informatics platform, which underpins all the data that we've collected and should help facilitate its harmonization and curation in future years. So nutrition and allergy throughout life is a very topical subject. This aspect was led by Kirsten Buyer from the Charité, with Thomas Kyle and Gideon Lack from King's, who many of you I'm sure have heard the work that he's doing. And it came out of the fact that we'd had this recommendation for not eating allergenic foods, and then we realised there was no evidence base for that, so that was withdrawal, and you're left with no advice at all. And we knew that there were a number of kinds of studies ongoing. What could we do to get better evidence? So the first aspect of this was to use our birth cohort set up in Europreval. And this is across different centres around the world, all the way from Iceland to Athens, from Spain up to Lithuania. And what this showed was the challenge that faces people with food allergies, but also faces the food industry is, I thought we were going to get one number out of this. We didn't. And what you can see here, this is cow's milk allergy in children under the age of 2. There's hardly any in Greece. Whereas if you go to the UK or the Netherlands, you're talking about over 1% of young infants having milk allergy. And it's not just milk. You see the same thing with hen's egg. So whatever's going down in Greece, there's much less food allergy there. And here you see the UK, up there, 2% of young children with confirmed allergy to egg. So what's going on there? And what's the differences? What's going on in Greece that's different to the UK? So we did a follow-up of the birth cohort through IFAAM. The children have now reached school age. And what you can see, the problem that faces all cohorts actually is that the number that you recruited and the follow-up, I'm afraid the UK didn't do very well. Our winners were actually, in the end, Iceland and Ireland. Iceland seems to have got, there we go, Iceland and Ireland with the baseline cohort. So overall, we got 58%, which isn't bad, but in the UK, it wasn't so good. What have we found out? Well, 73% of the children who had food allergies at 3 years, so this is far higher, they came back in to be evaluated. The good news, the vast majority lost their allergy. They're tolerant to milk or egg by the age of 6 to 10 years. Half of the young children with other allergies to foods like peanut, tree nuts, they'd also become tolerant. So that's also good news. However, 15% of the school children had at least 2 coexisting atopic conditions. These are the ones who are really vulnerable to food allergies. And actually, the potential risk factors were male, gender, and parental allergy. So the parental allergy, that's the genetic susceptibility. And I'm sorry, guys, but there's something about being a bloke that makes you more vulnerable. Which then Lynn's video, they should have a guy there because boys are far worse at doing medication than girls, I think. But maybe that's speaking as a mother of sons. So what about nutrition early in life? Interestingly, the majority of mothers initiated breastfeeding in the 9 centres, but the duration varied. Iceland had the highest prevalence of breastfeeding. Greece had rather low rates. So clearly, they don't have much allergy. They don't actually necessarily have as much breastfeeding. It must be something else that's contributing to the low allergy in Greece. There are differences, though, in the practices of introduction of complementary foods, so maybe that's part of it, but maybe it's also to do with the nutrition in those countries, things like latitude and vitamin D. So that's part of our ongoing data analysis. What we also did, because there were a number of interventions like the EAT and LEAP studies going on in Germany and also in Australia, so this is great because we have Kirsten from the Charité and Diane Campbell from Sydney. The Australians participated in our project, and that really confirmed that actually peanut allergy, the work that Gideon showed, that in early introduction in young infants seems to prevent development of peanut allergy later on in life. That was seen in other studies, so it's been replicated in other study centres. But actually there was no association at the introduction of egg or peanut allergy and the prevalence of allergy to that food. There may be problems with study designs and within individual RCTs, and certainly the egg does not seem to behave like peanut. We also looked at IgE sensitization, and in fact there was a differential effect on allergy and the IgE sensitization. So sensitization didn't change even though allergy was reduced. That's probably down to production of another kind of antibody called IgG4. So we're getting more insights into what's going on there. Risk factors and severity. So this was led by Ronald van Rey from Amsterdam with Montserrat Fernandez-Rivas and Lars Poulsen. And something that's been very important, work led by Montserrat Fernandez-Rivas, is to actually develop a severity score. Now, one of the things, this conference is about threshold, but of course, threshold's part of that story, but severity of reaction is also part of it. Is there a relationship between threshold and severity? Not proven, but maybe actually a higher threshold but having more severe reactions might be important in terms of how you manage an allergenic food. So we developed, first of all, an ordinal score, which is qualitative and easy for clinics to implement. But importantly, and for the risk modelling is important, is a numerical score, which has been developed using mathematical machine learning approaches with all of the data from Europreval. 8,000 different reactions were collected in that study, and we validated it, and severity was predicted in terms of emergency room assistance, hospitalisation, and things like medication. So other aspects that we've been touching on, and I know Shelley's going to speak this afternoon about the TRACE study, but in IFAAM we looked at the effects of the matrix on reactivity to peanut in a cookie matrix. We're waiting for TRACE to finish their study analysis before we can finish ours, because some of the patients are the same in Imperial. And we also undertook a drug intervention in walnut, and the preliminary data there, and it is preliminary, suggests there may not be an impact on threshold dose in one of the centres, but it might be that there is a difference in severity. And 25% of the population take some kind of antacid. So that may well be a significant finding. Taking all of that clinical information and feeding it into the risk models is very important. And it was wonderful to have Steve Taylor from the USA as a key partner in IFAAM leading this work, together with Astrid Kreutzinger, Jonathan Hurricane, and René Crevel, who I think has been instrumental in a lot of the study design that we've used in many of the projects. So this has developed a whole approach based on using the vital doses. Now, I know this is undergoing a review at the moment, but these are the candidate reference doses that Vital came up with. One of the things we did was a data gap analysis. And I think this is important to say, and there's been questions, how far do we go down the road of collecting more data? We have no data on things like certain tree nut species. We have hazelnut. We've collected more data on walnut, but many of these are missing. I don't think there's a threshold dose distribution for Brazil nut. Different fish species. Is it just cod? Is salmon as problematic? Crustacean, and we have no data at all on threshold doses for molluscan shellfish. Many of the foods we have data for less than 60 subjects. Now, when we started out of this, the back of an envelope calculation was you needed 30 patients. The confidence intervals are too wide. So actually, there have been studies done now. We know we need 60 subjects to get good data. And there are other things missing. What's the effective age? If you have a child or an adult, are they going to behave in the same way? At the moment, we're treating them as the same, but mass per body weight is very different in a child to an adult. We have an issue that maybe some of the data comes from single study centres, although that restricted geographic origin might reflect differences in patterns of allergies. So, for example, celery spice, we've got a lot of data from Switzerland, but that's because they have a lot of allergic reactions. Now, Lynn has mentioned how the Anaphylaxis Campaign were involved in IFAAM, and this is something that actually the patient organisations were very involved in helping us develop. It's the online reactions in the community study with Angela Simpson in Manchester and Audrey Dungalvin in Ireland. And this is just a snapshot. What we tried to do was to collect data on individuals having allergic reactions in real time, and in some instances we managed to collect food samples and analyse them. And this is just an example of somebody from Manchester. They ate a baklava, which had a label warning, walnut pistachio. They had a mild reaction, tingling in the back of the mouth and lips. Fortunately, this time, this guy didn't eat it. There were other occasions where he did, and he ended up actually having to use his adrenaline pen. It was just as well. When we got that sample and analysed it, it had a very high level of peanut. In fact, there was so much, if he'd had a 50-gram portion, he would have eaten nearly a gram of peanut protein. So whether that is cross-contact or whether it's substitution, we obviously don't know, but that's part of the data that we've been collecting and analysing and using with the risk models. We also tried to validate the dose distributions. Now, threshold doses are based on data collected in large groups of patients and then modelled. And you predict the level of allergen that might cause a reaction in, say, 10% or 5% of the allergic patient population. But we don't know whether that's really true. So actually, Steve Taylor, working with Katie, Alan, Wayne Schreffler, and Jonathan, came up with the idea of the peanut allergen threshold study, which is actually reported now and has validated the ED5 for peanut. We did the same study in egg and milk and hazelnut. We had a problem getting enough milk allergic kids. We nearly got enough for egg, but the hazelnut, we got a meaningful study size. We used 1 milligram of hazelnut protein, which is the ED05. We had 57 cases with 4 positive reactions in Spain, 62 in Switzerland, and 10 reactions in Switzerland. One of the patients in Spain suffered a severe reaction, 15% drop in FEV1. They were treated with salbutamol and steroids. So it shows that it supports the approach with PATS. This needs to be replicated with a larger multi-center study, but it also emphasizes that 1 milligram could still give you a severe reaction. One of the things I think we're going to need to assess is what is an acceptable level of reaction. I think the data from these studies will really help us working with the patients to work out, is that acceptable or isn't it? The IFAAM Food Allergen Management Toolbox was another big activity led by Astrid from the Netherlands, but with others. And this has really put together a toolbox which should be made available online once the papers associated with it are published. So first of all, there's an allergen tracking tool that enables you to say, is it likely that I've got cross-contact or contamination with allergen in a food? If yes, we then have something called the IFAAM Tier 1 risk assessment. This is slightly different to Vital in that it's based on food consumption patterns, not serving size. That's actually quite important because we know, dare I say it, in order to improve and cut the amount of sugar and so forth in the diet, portion sizes, are getting smaller. But unfortunately, when you have a teenage boy, sometimes they just go out and eat 4 or 5 of them. So you have to look at your 95% percentile. That's the sort of thing this does. Is the exposure greater than a vital reference dose? Yes. Okay, I go back round, I have to mitigate my risks. Are they enough? And you might go back round again. If they're not enough, you need to apply a precautionary label. It may also be that if it's yes and you've got a real issue, you do tier 2 risk assessment. This is probabilistic modelling. That's when you phone a friend and get someone like Joe Beaumont in FARP or Astrid and the team in TNO to help you. If you can sort it out, you don't have to do anything. But if yes, risk mitigation. And again, if you can't do it enough to get the levels down, that's the precautionary label. One of the things that's essential for that is knowing how much allergen you've got in your product. So this comes on to the next module, which is tools for allergen management analysis. And I'm not going to talk very much about this because I haven't got enough time, but we've done a lot on trying to develop multi-allergenic tools, immunoassay, but particularly mass spectrometry. And through the Manchester Food Allergy Network, we ran a multi-centre, multi-laboratory trial, which I have to thank the allergen test kit manufacturers who donated their kits and the labs who gave their time for peanut. And we showed very wide variations in method reporting, but somewhere between 19 and 80% of the dose, depending on which kit you used. There's obviously some work that needs to be done to improve collaborators and bring results together. The other thing that we actually had in this was all of our work developing the methods used foods and food matrices that were used for the patient studies as well as the development of the methods. Many challenges remain. I'm sure others who are going to speak will talk about this. Mass spectrometry, we don't have sequences of things like pistachio nuts and macadamia. That makes it more difficult. We lack reference materials and ways of calculating and reporting allergens. However, there is a real emerging consensus from the risk modelling. This needs to be in protein. Some of the methods don't, but it is possible for them to provide an answer in protein. That means you can use the tier risk 1 risk assessment in IFAAM or Vital. Lots of stuff we don't know about ELISA kits. I think some kit manufacturers are trying to address this. And mass spectrometry has a long way to go before it can actually deliver a reference or screening method. And we certainly demonstrated within the RING trial, it can be sensitive, comparable to ELISA's, but you have to have a modern mass spectrometry platform. Something that you've had for 5 or 6 years will not be enough, and that's going to require a lot of investment from analytical labs. Next steps to make PAL transparent. One of the things we did, again with the patient organisations intimately involved, Audrey Dunn-Galvin led them in doing a survey, looking at precautionary allergen labelling, and the conclusions of that was very much there needed to be a drive to get the food industry to adopt the use of risk assessment tools, such as IFAAM, tiered risk assessment and Vital. Reference doses are generally safe for the majority of allergic individuals need to be agreed, and they do need to be agreed by regulatory authorities to support the food industry. At the moment, we have a ridiculous situation in Europe. We have some of our regulatory authorities doing risk assessment using reference doses, and we have others who are operating a zero tolerance policy. Just be thankful that you're not in the USA because they have 2 different regulators doing it differently. So we have things that we need to assess. And I think the other thing from the patient organisations that came out of the survey was them, if there's no PAL on there, they need to know that you've done a risk assessment. And we really need to sort something out that if a risk assessment is used, it's done to a certain standard and it's indicated on the label. So coming towards the end, one of the things we know quite a lot about is how much is too much, particularly for foods like peanuts. And we know how low our methods need to go. Measuring how much, actually we've got some difficulties here, and we need to get much more reliable methods for allergen analysis to help inform the industry when to use PAL and to help the patients understand what PAL means for them. Is it safe to eat or not? Just to finish up, because treatment is in my title, and actually it's very interesting because one of the things that's changed in the last 10 years is treatments appear to be coming on the way. And that's for peanut at very least. And there are two companies in a race, DBV Technologies. They have a skin patch where they're applying peanut to the skin. And we have Immune with their Code It, where they're actually putting peanut flour into capsules. You break it and mix it in with the food. What we can see coming out of the data is DBV is a bit safer maybe. You don't have the reactions. You get itching on your skin, although some of the things I've seen look pretty sore. It works for children though, not adults. Don't know why. And it does induces tolerance to a degree. The oral delivery has side effects. People get constant allergic reactions. They may not be able to carry on with the treatment. There are also concerns over GI tract side effects. However, it's effective in children and adults, and it induces significant tolerance so that people can actually, in the end, tolerate grams of peanut. There are lots of others in the wing, pharmacological ones, subcutaneous therapies, oral immunotherapy with probiotics, boiled peanuts, there's a lot. But one of the things that's going to be important for effective delivery of the treatment, and my understanding is the DBV studies product and the Immune product both have fast track to get approval, so they go out to the patients quickly. But one of the things they have to grapple with are the outcome measures. And that means they have to demonstrate a significant improvement in quality of life. And interestingly, one of the things they're using is threshold dose. So they're showing, can people, after they've had the therapy, tolerate a much greater dose of peanut? And actually, does that mean they no longer have to avoid or worry about may contain? So I actually think now it's not just the food industry that needs these reference doses and thresholds, but actually the pharmaceutical companies who are going to deliver those therapies also need it. And that's very interesting because we can protect everybody through potentially having those. It will not only make life safer for those individuals with food allergies who can't have a therapy, but actually it's going to help companies get their therapies into the marketplace. And lastly, I just have this point is, okay, we may get a therapy for peanut, but we have allergies to all sorts of foods. And making therapies is expensive, and I think there are some challenges. DBV is a small company that started out with a lot of support from the French government. I think they're still an SME. Immune is a company that came out of the FARE group in the United States and a patient organisation. It is also a small company. So they're not going to have, it seems like they've got a lot of money, but they don't, relatively speaking. And I don't know what's going to happen about all the other food allergies. So lots of people have helped me with this, both in Manchester, Angela Simpson I've mentioned, and actually Katie Allen is a part-time appointment in the University of Manchester. My collaborators in IFAAM, Kirsten, Steve, Renee, Sabina, Jonathan, Barbara, Montserrat, we've worked for years. And lastly, of course, all those other partners in Europroval and IFAAM and the people who participated in our studies, allergic individuals who participate in a threshold study are very brave. The ones in the TRACE project, which you're going to hear about later on today, I don't know how they managed to commit themselves to do what they did with having multiple challenges. And just to say, the last one was important because we're going to have a meeting in April next year. Watch this space. Renee and I are working with ILSI and the IFAAM project team to actually disseminate our results. So Barcelona or Madrid, I think we're looking at. One of those. So thank you very much.
Thank you very much, Claire, for another wonderful presentation with such enormous clarity associated with it. Now, I believe we've got 1 minute, so I'm not going to let her escape. I'm holding on to her arm. One question from someone, please.
In your chart for the precautionary allergen labelling, your toolkit, you had acceptable risk. What is acceptable risk?
That is the $60 million question, isn't it?
That's why I'm asking it.
And one of the things that I'm a scientist and we produce data. I think what we've identified is a way in which we could understand what was acceptable risk using the data on the symptoms from the single dose challenges and to work with the patient organisations and their members to actually do some kind of online survey to actually get some data to say, are these levels of symptoms acceptable to them as allergy sufferers? And what's interesting in the peanut study, the PAT study, they only got subjective mild reactions. What I'm concerned about in our hazelnut study was in Spain, we had that one patient who reacted with really quite bad objective reactions. And in the Mediterranean area, they have these LTP type allergies. They're not the same as we get. And they have people having anaphylaxis to peach. So I think that would give us a vehicle for actually getting that information. And we know that that's what so for example, in Europe, it is DG Santé's job to define that together with the stakeholders, which in this case are the patient organisations. And I'm guessing, of course, we don't know where we are in the UK, but the Food Standards Agency is both the risk assessor and the risk manager for food, and it would probably be their job to work with the patient organisation, I'm looking at Chun, to actually help define what an acceptable level of risk would be. But I think subjective symptoms like oral tingling is less problematic. Having something like a drop in your lung function is problematic. And I think that there's probably a distinction there that can be made, but I think we need to get the patient organisations to work with us so there's a project there for somebody to fund. With Lynn.
Thank you very much indeed.